Welcome to USMLE High-Yields 2. Chromosomes and Genes. One of the most frustrating areas in the exam is remembering the name of the genes and chromosomes affected in certain diseases. These are actually the easy points in the exam because they are pure memorization. If you can memorize these genes and chromosomes, you will save a lot of time in the exam for other harder questions and collect these points. Ultimately, whether the question is hard and easy, it gets the same score. I hope these collections of genes and chromosomes on highly tested conditions will improve your score. So, let's master these once and for all. Wilms tumor. Wilms tumor is most commonly associated with a deletion of the tumor suppressor gene WT1 on chromosome 11. Huntington's disease. Huntington's disease. Gene, HTT, chromosome 4, anticipation, trinucleotide repeat disorder of CAG sequence. Job syndrome, also known as hyper-IgE syndrome. Gene, STAT3, abnormal neutrophil chemotaxis, decreased interferon gamma production by Th1 cell. Von Hippel Lindau. Von Hippel Lindau. VHF gene mutation leads to hypoxia-inducible factor, HIF, accumulation. Retinoblastoma. Retinoblastoma has RB1 gene mutation on chromosome 13. Marfan syndrome. Marfan syndrome. Gene, FBN1. Chromosome, 15, FBN1 codes for protein, fibrillin 1. Osteogenesis Imperfecta. Osteogenesis Imperfecta. Genes, CoIA1 and CoIA2. Type 1 collagen. Familial Hypercholesterolemia. Familial Hypercholesterolemia. Gene, LDLR, chromosome 19. Hypokalemic periodic paralysis. Hypokalemic periodic paralysis. Mutations in the CACNA1S or SCN4A gene. Achondroplasia. Achondroplasia. Gene, FGFR mutation, which is coded for protein, FGFR3. Adult polycystic kidney disease. Adult polycystic kidney disease. Genes, PKD1, PKD2, and PKD3. PKD1 is located on chromosome 16. Hereditary Spherocytosis. 5 genes affected in hereditary spherocytosis: ANK1, SLC4A1, SPTA1, SPTB, and EPB42. Approximately 50% of cases due to ANK1. For the USMLE Step 1, just remember ANK1. Sturge-Weber Syndrome. Sturge-Weber Syndrome. Gene mutation is GNAQ, located on chromosome 9. Tuberous Sclerosis. Tuberous Sclerosis. Genes affected are TSC1 and TSC2. TSC1, located on chromosome 9, code for Harmartin protein, and TSC2, located on chromosome 16, coded for tuberin protein. Neurofibromatosis, NF1. Neurofibromatosis has NF1 gene mutation, located on chromosome 17. Neurofibromatosis 2. Neurofibromatosis 2 has NF2 gene mutation, located on chromosome 22, produces myelin protein. Myotonic dystrophy. Myotonic dystrophy type 1 has trinucleotide repeat of CTG located on the 3' of the DMPK gene. But type 2 has trinucleotide repeat of CCTG in the ZNF9 gene. Familial adenomatous polyposis, FAP. FAP has gene, APC mutation, located on chromosome 5. 21-hydroxylase deficiency, also known as congenital adrenal hyperplasia, caused by the mutation of what gene? 21-OH, or CAH is caused by the mutation of gene, CYP21A2. Alkaptonuria. Alkaptonuria. Mutation of the gene, HGD. Ataxia telangiectasia. Ataxia telangiectasia, caused by mutation of the gene, ATM. Alpha-1 Antitrypsin Deficiency. Alpha-1 Antitrypsin Deficiency, gene, SERPINA1, coded for the enzyme, A1AT, located on chromosome 14. Beta thalassemia. Beta thalassemia, gene affected is, HBB, located on chromosome 11. Cystic fibrosis. Cystic fibrosis, gene, CFTR, chromosome 7. Chediak-Higashi Syndrome. Chediak-Higashi Syndrome, gene, LYST, list. Cystinuria. Cystinuria, gene, SLC7A9, or SLC3A1. Dubin-Johnson Syndrome. Dubin-Johnson Syndrome, gene, ABCC2, coded for protein, MRP2. Friedreich's ataxia. Friedreich's ataxia, gene, FXN, coded for protein, frataxin. Fanconi anemia. Fanconi anemia, genes, FANCA, FANCC, and FANCG. Galactosemia. Galactosemia, genes, GALT, GALK, and GALE. Gaucher disease. Gaucher disease, gene, GBA, coded for the enzyme, beta-glucocerebrosidase. GM1 gangliosidosis. GM1 gangliosidosis, gene mutation, GLB1. Tay-Sachs disease. Tay-Sachs disease, gene, HEX A GM2 Gangliosidosis. Sandhoff Disease. Sandhoff Disease, gene, HEX B GM2 Gangliosidosis. AB variant. AB variant, gene, GM2A, also GM2 Gangliosidosis. Hartnup disease. Hartnup disease, gene, SLC6A19, located on chromosome 5. Hurler Syndrome. Hurler Syndrome, gene IDUA, leading to a complete deficiency in the alpha-L-iduronidase enzyme, and lysosomal accumulation of dermatan sulfate and heparan sulfate. Hereditary Hemochromatosis. Hereditary Hemochromatosis, 2 genes, C282Y, and H63D. Krabbe Disease. Krabbe Disease, gene, GALC. Leukocyte Adhesion Deficiency. LAD, gene mutation, ITGB2. Do you remember what CD number associated with LAD? That's right. CD18. Very high yield. Maple syrup urine disease. Maple syrup gene mutations are BCKDHA, BCKDHB, and DBT. Don't forget the 3-branch chain amino acids associated with maple syrup: leucine, isoleucine, and valine. Metachromatic Leukodystrophy. Metachromatic Leukodystrophy, MLD, gene, ARSA, coded for enzyme, aryl sulfatase A. Phenylketonuria, PKU. PKU gene mutation, PAH, coded for phenylalanine hydroxylase. Sickle cell disease. Sickle cell disease, gene mutation, HBB, coded for beta-globin production. Wilson's disease. Wilson's disease, gene, ATP7B, located on chromosome 13. Alport Syndrome. Alport Syndrome, genes, COL4A3, COL4A4, and COL4A5. Type 4 collagen. Rett Syndrome. Rett Syndrome gene, MECP2. Vitamin D-resistant rickets. Vitamin D-resistant rickets, also known as hereditary hypophosphatemic rickets, has a gene mutation, FEX. Fragile X syndrome. Fragile X syndrome, gene, FMR1 Triple repeat CGG nucleotides. Ornithine transcarbamylase deficiency. Ornithine transcarbamylase deficiency. Gene, OTC. Duchenne-Becker muscular dystrophy. Duchenne-Becker muscular dystrophy. Mutation gene, DMD, leads to the alteration of protein dystrophin production. X-linked intellectual disability, XLID. XLID gene mutation is, PHF8. Glucose 6-phosphate DH deficiency, G6PDH. G6PDH gene mutation, G6PD, which is coded for the enzyme, G6PD hydrogenase, to convert glucose 6-phosphate into 6-phosphogluconate to yield, NADPH. NADPH is needed by red blood cells to fight free radicals and for the production of the reduced glutathione. Leigh syndrome. Leigh syndrome. Gene, SURF1, coded for protein, COX. Myoclonic epilepsy with ragged red fibers syndrome. MRF. MRF gene mutation, MTTK. Turner syndrome. Turner syndrome gene mutation, SHOX, SHOX, on X chromosome. Cri-du-Chat syndrome. Cri-du-Chat syndrome caused by sporadic mutation, the partial deletion of chromosome 5. Remember the high pitch, cat-like cry. Pottau syndrome. Pottau syndrome. Trisomy 13. Edwards syndrome. Edwards syndrome. Trisomy 18. Caused by non-disjunction. Down syndrome. Down syndrome. Trisomy 21. Majority of cases caused by meiotic non-disjunction. Mosaicism is most common in Down syndrome. DiGeorge syndrome. DiGeorge syndrome. Gene, DBX1, chromosome 22. Remember catch-22. C is for cardiac abnormalities, A is for atypical facies, D is for thymic-aplasia, C is for cleft palate, and H is for hypocalcemia and hypoparathyroidism. Prader-Willi syndrome. Prader-Willi syndrome. Caused by the paternal deletion on chromosome 15. Angelman syndrome. Angelman syndrome. Caused by the maternal deletion of chromosome 15. Thank you for watching. Please click like, subscribe, and share.